Well, thanks for everybody that's joined. I'm going to talk about Genie this lesson. So last week, Ben gave a really nice talk about annotation using HMMs, and I thought this would be a nice follow-up to that lesson. Genie is a program that I wrote over the last year and a half, and it is an HMM-based program in that it is a series of custom scripts, and it uses a library of HMMs that some of which I've created and some of which I took off from Tigrafams and Pfam. And I'll talk a little bit about Pfam and Tigrafams a little later on in this presentation. Just a quick note, if anybody has questions, feel free to ask in the chat. Once I figure out how to work the chat while I'm giving the talk. There it goes. Okay, so I have the chat open. So if you have a question at any point, feel free to chime in, and then I'll do my best to answer it. Genie is a program I wrote with Nancy Marina. So Nancy is actually deserves a lot of credit for this. I probably should have included the authors list, but Nancy was actually the one that came up with the idea of having a tool that focuses on iron after she dealt with trying to annotate a bunch of metagenomes, and she found it really difficult to really find and categorize all the iron-related genes in those metagenomes. So she came to me and she had this idea: why don't we make a systematic sort of approach to categorizing iron genes, and then make HMMs and try to put them in the tool that will profile an assembly or a genome with those HMMs. So Nancy deserves a lot of credit, and also Ken Nielsen was a great help with this. So what is Genie? So Genie is it's a collection of HMMs. It's based on a comprehensive set of genetic markers, and it's sort of, we try to be as comprehensive as we could with regard to anything that's related to iron acquisition or scavenging. And when I say scavenging, I mean siderophores. So siderophores are these molecules that cells excrete that bind iron, and then they encode transporters to selectively transport these molecules that have bound iron. Transport, efflux, and storage. So storage is primarily ferritin. And one of my favorite things that's included here, and this is because it was the subject of my master's thesis, is the iron redox. And that's something that I'm really interested in, is thinking about how cells are actually able to use iron as a source of energy, whether to use it as an electron sink, where they can get electrons out of hydrogen or organics and then dump them onto ferric iron, or whether cells can extract electrons from iron too. So basically, Genie is most of the effort was creating this collection of HMMs, and then we compile this together and put together a bioinformatics software that uses these HMMs to profile genomic data sets. So as I said, these can be metagenomes or genomes, and they could also be transcriptomes if you've reassembled the transcripts. I'll talk a little bit more about this, but it uses rules for filtering out potential false positives, and that's based on the known operon structures that's involved in iron acquisition. So if you're dealing with iron transport, for example, that there's a certain set of genes that are typically encoded in an operon, like a permease and an ATPase and those kinds of components. So it uses those known operon structures like in the case of the similatory iron reduction, Schumanella encodes the MTR pathway. So that's sort of a known module of 3 or more genes. So you have a periplasmic cytochrome, you have a pore, and then you have an outer membrane cytochrome. So knowing that, the scripts that are involved in Genie sort of take that into account and filter out potential false positives. So if you have a decaheme cytochrome without any porin, that's sort of that's weaker evidence that there's something iron reduction related going on. Right, and then bit scores is another thing, and I'll talk a little bit about that. Okay, so this is a table from the Genie publication. This is not something that I really wanted to include, but I just wanted to sort of showcase the fact that we have HMMs from all over the place. We have HMMs from Pfam, we have HMMs from Tigrafams, and the ones from Pfam or Tigrafams are bolded and underlined. And the ones that are not, the ones that are not bolded or not underlined, are the HMMs that we made specifically for Genie. So I kind of wanted to do a very brief segue into what I mean when I say the HMMs are from Pfam and Tigrafams, for those that might not have experience using HMMs from those sources. And this is something that I had to figure out. And I kind of wanted to just do a brief kind of a, not a tutorial, but just sort of a showcasing of how easy it is or how easy it can be. So this is the Pfam webpage. So it's really easy to find. You just type in Pfam, and for me, Google auto-completes it. But if it's your first time going on Pfam, you might have to find this. There's a really nice keyword search. So if you have a gene that you're interested in, like for example, I typed in rhodopsin, and then the first result that came up was this first Pfam, actually, the first Pfam family. There's thousands of these Pfam families, and these are domain-specific families. So if you download an HMM, the HMM is going to be specific for a certain domain, not a gene. So that's one thing to keep in mind. So since I was interested in rhodopsins, I typed in rhodopsin. I got this hit, and I clicked on it, and it's a 7 transmembrane receptor domain. This is a solved structure of it. It's really nice. So you can just click on that and you can explore the structure. If you want to use the HMM for this, what you would do is then you would, how does this work? Then you would click on curation and model. And that will take you to this page right here. And in this page, you click the download link, and then the browser will automatically download the HMM for you. There's some useful information on this page. One of them is these cutoffs that they provide for you: the gathering cutoff, the trusted cutoff, and the noise cutoffs. I'm not going to go into too much. I'm not going to go into what the differences between these cutoffs are. I usually go with the trusted cutoff. Actually, I usually just go with the highest, just to be conservative when I'm doing my HMM searching. This is what the HMM file looks like once you download it. So in the file, if you download an HMM file from Pfam, it's going to have these 3 lines in it, and these will actually mirror exactly what I just talked about here: the gathering cutoff, the trusted cutoff, and the noise cutoff. So when you run your HMM search, sorry, this is kind of a lot to take in, but and actually on Wednesday I'll be going over a lesson on how to make your own HMM and how to use HMM search. So if you want to tune in on that, but just to sort of briefly mention here is that once you get this HMM and you want to use this HMM to search against the protein data set, you can add this -- cut TC flag, and it's going to automatically use this trusted cutoff setting when it's using the HMM to search. Okay, so that was that was my brief segue into Pfam. Tigrafams is another great resource. I really like these HMMs. They're not really adding more HMMs to this resource anymore. But the HMMs that are there are still available. And you can find it if you just do a Google search of Tigrafam's complete listing. And that should take you to this page, which has thousands of HMMs. And these HMMs, as opposed to Pfam, they're not families, they're not domain specific, they're gene specific. So if you want to use these HMMs, what you want to do is you click on any one of these. So for example, if you're interested in trying to find ribosomal protein BL35 in your protein data sets, click on that. That'll take you to this page, and you'll see you'll have a similar trusted cutoff, noise cutoff, and domain trusted cutoff. And then you'll click on seed alignment. That'll take you to this page, and then you can download the alignment. And then once you have the alignment, you can build the HMM, and then on Wednesday I'll talk about how you can go about doing that. Okay, so that's it. That's my brief segue. I'm going to get back into Genie now. So basically, there's over 200 HMMs involved in Genie, and these HMMs are grouped into 5 broad categories, which are also broken up into smaller categories. So you have iron acquisition, and this involves anything that a cell can do to get iron. So you have iron-ion transport, so therefore synthesis and transport, and you also have bacteria that are able to obtain iron from heme. So they will import the heme in and break it open and basically get the iron from that. So you have iron gene regulation, iron oxidation and reduction, iron storage, and magnetosome formation. So
Arkady, you have a question in the chat box.
Oh, right. Thank you. Let's see. Barbara Bayer is asking, what are thresholds for a good cutoff? That is a good question. And that's actually, that would take me a little while to explain. And I'll actually talk more about that on Wednesday, exactly what goes into setting a threshold for an HMM. And it's kind of a complicated process, but essentially what you want is you want a threshold that will identify all the genes that are part of the family. So if you make an alignment, so I'll go into this on Wednesday, but if you want to make an HMM is based, and actually Ben Tully talked about this last week, so if you want to look at functional annotation lesson 3, I think that might actually help you out with understanding actually what goes into making an HMM and how thresholds play into that. But actually, a lot of work went into figuring out exactly what cutoffs we should use for these HMMs, and it's kind of a difficult thing to figure out. Essentially, when you make an alignment and you use that alignment to make an HMM, you would want the, and then you make that HMM, you use that HMM to search back against the alignment that you used to make that HMM. You want to take the lowest bit score of the results that you get back. And if that bit score doesn't hit any false positives when you search it against a bigger database like NCBI's NR database, that would be considered a good cutoff, in my opinion. But it doesn't always work out that way. But yeah, I don't want to segue too much into that because that's sort of a whole lecture in and of itself. That's a good question. Thanks for asking it. Yeah. Okay. So I think that's it for this slide. Oh yeah, I'm not going to dwell too much into the breakdown of iron oxidation and reduction, except to mention that there's really not a lot known yet about mechanisms for iron oxidation and reduction. and the genetic markers are putative, and more so, some more so than others. Like there are some like MTOAB that's under the probable iron oxidation, possible iron reduction category, and that's because this has been suggested to be an iron oxidase in some organisms, but in other organisms like Shewinella, homologs to this gene are actually called MTRAB. You can see they're part of this operon, and they're actually involved in iron reduction. So there's a lot of ambiguity to this, and that's why we decided to really break down iron oxidation reduction into 4 categories rather than 2 categories, just to sort of highlight this ambiguity. Okay, so this is a figure from the publication. Basically, just showcases the various ways that bacteria can obtain iron, and you have lots of different mechanisms for it, a lot of different components. These are very diverse pathways. I'm not going to talk too much about this. If you're interested, you can check out the publication. The iron scavenging, acquisition and scavenging strategies are actually very important in iron-limited environments, and we actually show this in the article because we used a set of metagenomes from various ecosystems, including the open ocean, which are all here on the left part of this dot plot. And the rest of the dot plot is sort of dealing with environments where there's more likely to be a higher concentration of iron. Like a river plume represents a place where a river empties out into the ocean. So you are more likely to have more metals in sort of that kind of system. Hydrothermal iron mat, that's as iron rich as you get. And then you have groundwater aquifers that can also have high concentrations of iron. So in these sort of categories that are related to iron acquisition, you actually have more genes involved in that. Yeah, it is really cool. Thanks, Adelaide. I think it is really cool to kind of see it this way. We do see, consistent with what we know about hydrothermal systems, we see potential for iron oxidation, quite a lot of potential for iron oxidation. So these are data sets they actually got from Sean McAllister, who won on all these cruises to the Lewicki Seamount and the Mid-Atlantic Ridge, and they collected these just really thick iron-rich iron mats, so iron microbial mats, and then he sequenced them. So this is all of his data that he recently published on, and he was nice enough to send me the metagenome assemblies. And yeah, there's a lot of zeta proteobacteria there, and zeta proteobacteria are obligate iron oxidizers. So it makes sense that we find a lot of iron oxidation genes. And we don't see a lot of them everywhere, but we do see potential for iron oxidation in environments where it's not really expected, like the surface ocean. We see genes that are involved in iron oxidation and reduction. And it's not really clear how they're, there's really not an apparent source of iron that would be enough to sustain iron reduction. So that's something that's, I think, is mysterious to me, at least. Potentially there's particulates that could be sort of seeded by iron reducers or iron oxidizers. Okay. I should mention that for the siderophore heme transport, and we sort of highlight this heavily in the paper because it is a significant limitation of Genie, is that these genes that are involved in transport of siderophores, that's known as the TON-B dependent receptors. It's a 3-gene operon. And the issue with it is it's not exclusive to siderophores. So the TON-B dependent receptors are able to transport a wide variety of substrates like sugars and basically things that might not be related to siderophores. So that's why it's explicitly stated as potential for siderophore transport. One of the things that we're actively working on right now, the Genie team, is to try to figure out how we can better discriminate between TON-B dependent receptors that are specific for siderophores and heme and those that are not. But the current version of Genie doesn't really discriminate between the different TON-B dependent receptors. So that's why this is a potential for siderophore transport. It doesn't necessarily mean that that's exactly what all the TON-B dependent receptors reported here do. Actually, it's likely not what they do, but okay. So just briefly, the iron oxidation and reduction. Basically, the key takeaways is that porin cytochrome modules are very important in this case. So porin is an outer membrane protein, basically forms a pore, and cytochromes are redox active proteins, and they bind heme, and heme is the active site of that protein. So you have various numbers of heme per cytochrome. So you have decaheme cytochromes. Geobacter include some astounding number of hemes in their cytochromes. I think the biggest I've seen is a cytochrome with 26 hemes in it. Recently, there was actually a paper published where they show how these hemes are organized in Schuenella's MTRC protein, and it's really cool. Actually, it's a molecular wire. The reason these bacteria need to do this is because iron oxidation and reduction needs to occur outside of the cell, and that's because iron 3 is largely insoluble. As soon as iron 3 is produced, for example, in iron oxidizing bacteria, they oxidize iron 2 becomes iron 3, it's immediately precipitated as iron oxides. You don't want that reaction occurring inside the periplasm of the cell. You want that reaction outside of the cell, because if you have stuff precipitating in your periplasm, you're not in good shape as a cell. So you want to keep that reaction as far away from the cell as possible. Iron reducers have to deal with a different problem, and they're dealing with something that's already solid that they can't import into the cell. So what they need to get reducing equivalents away from the cytoplasm membrane out into the extracellular space. And the way they do that is these porin cytochrome modules. There's also the Geobacter includes some other mechanisms that aren't really porin cytochrome related. For example, OMCS, polymerized OMCS. So OMCS is a tetraheme cytochrome, so it has 4 cytochrome groups, and it actually forms polymers, kind of like pili, these sort of long chains of OMCS that extend out from the cytoplasmic membrane out into the outside of the cell. And the way that the heme groups of these cytochromes link up, that should allow for electrons to actually get out from the cytoplasm to iron that's outside of the cell. That's actually a really cool mechanism that was published just last year. Another thing that's not really included here are the pili, and that's something that's Jennifer Glass mentioned in the Slack page, is that you have these pili that are enriched in aromatic amino acids, and the way the aromatic side chains of these amino acids line up, they would allow electrons to travel for a long distance, distance long enough to get electrons from the cytoplasm to the outside of the cell. Okay. And this is just basically showing the iron cycle. One of my interests is figuring out the syntrophic relationship between iron oxidizing and iron reducing bacteria, and how this sort of cycle plays out in the environment. So this is the general workflow of Genie. It's basically, like I mentioned, it's a combination of custom scripts, and it also wraps several programs, including Protocol, BLASTP, HMM search, and there's also Diamond, but this is an optional sort of cross-validation step that I recommend, but you don't have to do it if you just want a quick and dirty look at the potential iron genes in your data sets. So just to briefly go over this, the input is a folder of genomes or metagenome assemblies. If you have just one assembly, you have to put it in a folder, you have to provide the folder. That's just how it works. And so that's the input. And you can provide an assembly or you can provide gene calls. So if you provide an assembly, Genie will run prodigal for you and it will predict genes from the assembly. If you provide gene calls, it won't do that. It will just go straight to the BLAST P and the HMM search. So the HMM search uses the HMM library of iron genes, and the BLAST P blasts a collection of genes for which I wasn't able to make HMMs for one reason or the other that I'm not going to get into now. But that's explained in the paper why. So then these results are clustered based on genomic proximity, and the results are summarized, and potential iron oxidizers and reducers are also identified based on the presence of iron redox proteins. So this cross-validation step I put in because I think that, like all annotation methods, there is a chance for false positives. So if you have a protein database that you trust, like NR or RefSeq, you can provide that, and that'll be in part of the tutorial. I'll show you how to do that. It's just an additional flag when you run the command. So when you do that, what will happen is that every potential iron gene that's identified by Genie will be diamond blasted against this protein database. And then when you get the spreadsheet, the CSV output, there will be an additional column in that spreadsheet, and that column will include the closest blasted from this reference database. That's essentially the main output. I'll go over this during the tutorial, what that looks like. You'll get the heat map. This is a really nice summary of iron genes. The heat map is basically used to generate this dot plot that I showed. That's also an optional. You can, once you get the output, the spreadsheet, you can plot it however you'd like, but there's an option to have Genie run R script for you and generate the dot plot. Okay, one more slide and then we'll get to the tutorial. So this is basically the validation that I ran of Genie, just to see whether it identifies the known iron genes and bacteria that are known to encode those genes. And we do see that for the redox, we have the iron oxidizers here, Rhodobacter, Mercurifundus, the zeta proteobacteria, heteroxidants. So they all encode iron oxidation. The iron reducers encode iron reduction genes. You have some possible iron oxidation reduction, and that's the case for Rhodopseudomonas palustris. And like I mentioned, Rhodopseudomonas palustris encodes the MTOAB pathway, and there's ambiguity in that pathway. So that's why it's labeled as possible iron oxidation reduction. Yeah, there's the siderophore producers that make siderophores, and there's the iron-free bacteria, which I sort of a last-minute addition. They're not really iron-free. They're actually symbionts, and it's really not known how they are able to get iron, but Genie did identify some potential transporters and a gene that's potentially involved in iron gene regulation. So the jury's still out as to whether there are actually truly iron-free organisms out there. Okay. That's it. Well, right in the dot, 30 minutes.